Hi, this is Dr. Lauren Launen, and this is the first of a lecture set on DNA synthesis, also known as DNA replication. In this lecture set, we will also discuss telomeres. The goal for this lecture set is to discuss DNA replication with the addition of sophomore level detail, i.e. the names of genes and proteins, and a more sophisticated understanding overall of DNA synthesis than what you would have had in your first year. We'll also focus on telomeres, what they are, why they're necessary, and how they are synthesized. So DNA replication happens during S phase in the eukaryotic cell, and in a prokaryotic cell, it simply happens just prior to replication or binary fission. It's a process that occurs using a strategy called semi-conservative replication. And what that means is you utilize the double-stranded DNA molecule as a template. So here at the top of this figure, we have the double-stranded DNA molecule and the characteristic double helix shape we've discussed elsewhere. Each of these two pink strands will be used as the template for the synthesis of new strands shown here in blue. So what you get after each round of DNA replication is from one molecule you make two molecules, and each of those two molecules is half new and half old, and that's the idea of semi-conservative replication. Something that was determined through a set of experiments by scientists Michelson and Stahl, which we will discuss in class. So in order to make a new molecule of either DNA or RNA, you have to utilize a polymerase enzyme. You use DNA polymerase if you're making DNA, you use RNA polymerase if you're making RNA. Shown here is a figure that focuses on the growth or extension of one single strand, one new strand of nucleic acid. In this case, it's DNA. I know that because there's no hydroxyl group at this position, and also the word DNA polymerase is written here. So here we've got a growing strand of DNA. So, and this is showing the new nucleotide triphosphate in blue being added to the 3' carbon on the sugar of the preceding nucleotide. So all of this here is a nucleotide, right? And then all of this here is a nucleotide triphosphate. And it is being incorporated into this growing strand. We are growing this strand in the 5-3 direction. So DNA polymerase is a pretty cool enzyme. It can go really fast. It doesn't make a lot of mistakes. It's a pretty amazing product of evolution, of many millions and billions of years of evolution, but it does have some limitations. And one of those limitations is that it can only catalyze the attachment of this nucleotide triphosphate at this particular position, okay? So in order to do this, what you're going to need is nucleotide or nucleoside triphosphates, and this is a nucleoside triphosphate shown here, 1, 2, 3 phosphate groups. You're going to need the cofactor magnesium, and that's so that the DNA polymerase will work. It turns out that DNA polymerase, like RNA polymerase, needs to have a little bit of magnesium attached to it, or it is non-functional. You'll need a set of other proteins for this to work in the cell, and we'll talk about that in a moment. And you will need a template strand, and the template strand is not shown here, but the template strand will play a role in base pairing, and that will control or dictate the identity of each new nucleotide as it is added to this growing strand. You also need the right environmental conditions. So generally you need to be at a fairly neutral pH, and whatever the appropriate temperature is for the DNA polymerase that you're working with or that you have inside of a cell. So a little bit of history here. Here are two extremely famous scientists, Severo Ochoa and Arthur Kornberg, and they were awarded the Nobel Prize in Physiology or Medicine in 1959 for their discovery of the mechanisms in the biological synthesis of ribonucleic acid and deoxyribonucleic acid. And what Ochoa and Kornberg did is each of them working independently with bacterial organisms, Kornberg with an E. coli and Ochoa with an acetic acid bacterium, they isolated the respective polymerase enzyme responsible for the synthesis of either DNA or RNA. Ochoa isolated RNA polymerase, and Kornberg isolated DNA polymerase, something we now call DNA polymerase 1 in order of discovery. So both these men, in their groups, they isolated these proteins, and then they used them to synthesize either RNA for Ochoa or DNA for Kornberg. So they figured out the conditions needed to have synthesis, and they were the conditions that I just listed for you in the previous slide. You need to have a template, you need to have nucleoside triphosphates, you need some magnesium, you need the polymerase enzyme, and you need the right environmental conditions. And so that was some pretty important work, and for that they were awarded a Nobel Prize. All right, so DNA synthesis involves more than just the polymerase enzyme itself, however. So here is a slide that focuses in on DNA synthesis in bacterial systems, and that's what we will focus on in this class. So look at this list of proteins, right? 1, 2, 3, 4, 5, 6, 7 proteins, each with a different role, and all of them are necessary to have DNA synthesis work in a cell. So just to orient you a little bit on this figure, here's the double-stranded template molecule, and the new DNA is being shown in blue, just as in the previous figure. And we've got some extensive labeling going on here. So let's look at where the different components are. DNA topoisomerase, which is also frequently called gyrase. Gyrase, topoisomerase, same thing. It's shown here. Its job is to relax what is called supercoiling so that the double-stranded molecule can open up, creating this opening, which is called a replication fork. So you can't have that without topoisomerase traveling ahead and relaxing the tightly coiled DNA molecule. Next we have an enzyme called helicase, okay, and the gene for that is DNA-B. Its job is to actually unwind the double helix. So in order to unwind it, what you're going to have to do is you're going to have to break the hydrogen bonds called base pairs that are holding this double-stranded molecule together, and that will then allow formation of the replication fork, shown here. We have a need for enzymes, or not enzymes, but proteins called single-strand binding proteins, SSBs, or sometimes they call them SSBPs. And they are shown as like these little purple dots. And their job is to prevent reannealing of the separated strands. So when you break those base pairs through the activity of helicase, the strands would like to just base pair again. That could happen spontaneously if you let it. So kind of like putting gum in a zipper would prevent you from zipping up the zipper, annealing these single strand binding proteins onto the single strands at this moment in time will maintain the replication fork, keeping it open. The enzyme primase is responsible for the synthesis of an RNA primer. And so primase in this figure is shown, here's the RNA primers shown in orange here and here. And let's see where primase is. Primase is actually not shown on this figure, but it is responsible, for making these primers. So it was there a moment ago. All right, so then we have two DNA polymerases shown. DNA Pol is short for polymerase 3 and 1. DNA polymerase 3 is what does the major synthesis in all bacterial systems. And it is shown in this sort of blue blob here with this little clamping loader part keeping it together. And DNA polymerase 1 is not shown actively in this slide, but we'll get to it later. Its job is a little more focused. It's going to remove and replace the RNA primer with DNA later in synthesis, a moment later. And then finally, we're going to need the enzyme DNA ligase. And it's going to join DNA segments together. That's going to happen primarily at what is called the lagging strand. So you don't get DNA synthesis without the activity of a lot of different proteins. All right, so let's focus in for a moment on the DNA polymerases that are found within bacteria. So Arthur Kornberg, as I mentioned, discovered DNA polymerase I. And as I said a moment ago, DNA polymerase I's activity is necessary to replace the RNA primer with DNA at the primer regions on the new strands of DNA that are being formed. DNA polymerase III is necessary for doing the majority of the work in DNA synthesis. And then there's also DNA polymerase II. And in addition, there are polymerases 4 and 5. And 2, 4, and 5 typically play roles in DNA synthesis that happens as part of DNA repair processes. So we're going to focus chiefly on 1 and 3. And each of these is able to catalyze the formation of a phosphodiester linkage. That's really important, right? That's what DNA synthesis is all about, is formation of phosphodiester linkages. But each of them, while it has that in common, has some differences as well. So none of them are able to initiate chain synthesis. In other words, each of them has to have a 3' carbon with an OH on it from a previous nucleotide in order to carry on with synthesis. They can't start it new or de novo. They can all make phosphodiester linkages. That's what this polymerization activity is. And they all do it traveling 5' to 3'. That's what I was showing you on an earlier slide. This 3 to 5 exonuclease activity, and similarly 5 to 3 exonuclease activity, exonuclease means to break phosphodiester linkages. So all of them are able to travel 3 to 5, which is backwards, and break phosphodiester linkages. And that'll be important in DNA repairs we'll talk about later in this class. Some of them can also do that moving 5 to 3. DNA polymerase I is able to do that. So it's a little different from the other DNA polymerases in that regard. The molecules of polymerase per cell, so how much of this protein is in each cell, there's a lot of DNA polymerase I, and there's a smaller amount of DNA polymerase III, which might seem odd, but it's enough to allow the cell to do its work. All right, remember that DNA replication is going to utilize both strands of a template molecule at the same time, right? So that means that you're having two actions happening at the same time, and they need to be coordinated in space and time very precisely. So this is a diagram from a Nature review of DNA polymerase III from E. coli, right? And this is typical for all bacterial DNA polymerase III. And what you notice is the symmetry of this molecule, okay? So one side will make DNA according to one of the strands in the double helix, and the other strand will make new DNA according to the other strand, right? And then you've got these coordinating molecules in the middle. And so each of these little colored blobs is a protein subunit. This is an example of a protein that definitely has quaternary structure. Everywhere we see one of these symbols, there is a gene that encodes that particular polypeptide that forms this particular protein. We'll have to express that gene once and twice to build an intact DNA polymerase molecule that the cell can then use. And these are the functions and names of all of the different subunits that we just looked at. So the gamma complex is going to load the enzyme on the template, like get it onto the strand. The alpha, epsilon, and theta complex are going to actually do the phosphodiester linkages. They're going to do the catalysis, and they're also going to do something called proofreading. The beta clamp allows the DNA polymerase III to slide along smoothly and not skip over any areas. And then this tau unit holds the two sides together. It does something called dimerizing the core complex. In total, DNA polymerase III is a great example of what is called a holoenzyme, which means it has multiple parts that all must come together in order to get a functional protein.